Hey everyone, thanks Chris. So, slide. It's a pretty simple story I want to try and get across to you all today, and that's mainly about some of the tools that we use. So it's come up in the videos that there's tools like the next generation sequencing and genomics, which really opens the door to characterising the detail of the microbial diversity out in the different environments. So when microbiome science with genomics meets restoration ecology, which is only a recent meeting place, we can not only document the recovery of ecosystems through traditional methods of monitoring plants or animals in those ecosystems, but these two graphs here show in a mining context and a post-agricultural context that you can get a microbial restoration occurring in those landscapes as well. So what that means is we have the tools and the evidence then to know that we can restore microbiomes, environmental microbiomes. But then the question would be, can we actually do this in urban settings where the challenges are very different? So we've applied this restoration toolkit to urban green spaces and have shown the same thing. So basically, when you do urban green space restoration, so you're replanting native plants, you're managing the native vegetation and animals and dealing with the invasive plants, you get a concordant response to the microbiomes in those environments. So why is that important to HUMI? That's the next slide. So what that means is, and it's come up in the videos as well, so when we go out into those green spaces, what's actually happening to us? And there's some good theory discussing that we should be bombarded and colonised by the environment, but you can actually measure that using this microbiome science. And the big graph on the right-hand side there shows that. In blue, we took some samples from people's skin before they went out into urban green spaces, and then in yellow, what happened to their microbiomes after exposure. And what that shows is we're getting a substantial shift. And so what that means is the environmental microbiome can be restored and it colonises us, and we know that there are close links between the human microbiome and our health, as we've heard through the videos and we'll hear more about in the talks coming up. So just to rehash, we've got the tools and evidence to know that we can restore environmental microbiomes. We can do that in urban green spaces, and we get colonised by those environmental microbiomes. That's what I've got. Any questions? Yeah, so the question was how much exposure is required to observe a shift in a human microbiome, and how long does that persist? So that slide there shows a couple of people in the environment, and they were exposed for, some of those yellow dots were just 20 minutes, some of those were an hour, and so we're observing a very rapid shift in the environmental microbiome onto humans. And the key point is those blue dots are the same people measured across different days. So absolutely, we're getting a resetting of the human microbiome through time. Any further questions? Sure.
When you talk about microbiome, you talk about microbial flora only, or microbial flora plus microbiome from outside, or contaminated flora?
Could you just repeat that again, please? Sorry. Can you repeat that again? Can you repeat your question, please?
Oh, yes. When you talk about microbiomes, okay? You mean only microbial flora observation, or microbial flora observation plus microbe come from outside?
Ah, okay. Sure. So, yeah, I'll repeat that one. So the question, I think, it's a complex of...
we are in the city, people come from outside, so we bring with them other microbes, which is not indigenous microbe in the area.
Okay, so the microbiome is a way to characterise all the microbiota from an individual sample, right? And in this case, these samples were, this example up here, that's from a skin swab of people. So I think your question related to what the origin of that changing microbiome is, whether it's from the local environment or from far afield. Is that right? So in this case, we know that when you look at the details of these microbes, that we also sampled from the environment in these different spaces where the people went, and there were particular species of bacteria in this case that came from the environment, so from the soil, from the air, where these people were exposed. So I think there's still a lot of science to be done, but we know that there is indigenous sort of local environmental microbiomes that colonise us. And whether that is, whether there's a great diversity of those microbiomes out there that we haven't sampled and we don't know the function of, absolutely. But we can definitely say that there are environmental microbiomes that are colonising us. I hope that answered your question. I'm happy to catch up during the breakout session to discuss in more detail.
I'm going to bring around this mic. Here you go.
Thank you. I have two questions. So one was to pick up on the original question there. You said you used the word reset there, as in the microbiome is reset, but for how long does that effect remain? Because that would, for policy implications, that would be quite significant. Do we just have to do this once, or do we have to get people out there every month, or what's that? And then my second question relates to, you said that the microbial community is restored with, and you've got evidence of that. There's a clear change in the microbial community. Do you have any idea of how representative that new community is of what you would expect naturally in a primary habitat that was unaltered? And is there a need for us to also consider reintroduction of microbiome communities or restoring ecosystems?
Both very good questions. I'll go to the second one first, and on both of those slides, it's a bit difficult without a laser pointer, but we had reference ecosystems where we could track then the restoration of these microbiomes towards that reference ecosystem. So through the traditional restoration pathway of replanting and controlling invasives, the microbiomes are returning to a restored state. So the blue dots really in all those graphs are showing the microbiomes from remnant or reference ecosystems. But I think your first question perhaps is more intriguing, and that gets into the molecular epidemiology side of things, and that's not my area of expertise. But my understanding is when you get colonised, some of those microbes are going to potentially be ingested by us. So that was a skin swab sorry, if you go forward another slide. So those swabs there are from our skin on our wrist, but we know that there's transfer from things that colonise us into us. And so it's plausible, and I think there's probably other people in the room here who can talk about the more direct human health implications of getting colonised. So this information is purely about what's on our skin, and you're right, it does reset, but we would expect some of those colonising microbes to be ingested, to be breathed in, and that's where the effect onto the internal human microbiome, I think, would take place.
Hi, just before you ask that next question, let me just add to that slightly. So we've got 30 to 40 years of green space and human health studies, which basically show that the closer you live to an urban green space, the better your health is, at a population level, right? Not at individual. We're talking about groups of people. And so that's very consistent in most of the literature, particularly for the better studies. What is lacking is the mechanism. We don't really understand why that should be, okay? So when you walk into a park and we're measuring and shifting your microbiome, we're pretty sure that that has to be on a frequent basis, right? It's in the air, right? This is an aerobiology, you're breathing it. So a one-off shot probably isn't gonna do it. But actually getting the dose response, that's the holy grail. And I think that's really where we're heading, because until you can actually measure that and say, okay, it's 3 hours in a park that has this kind of macrobiology, biodiversity, the health sector's not going to take it seriously. So that's where we're heading, and it's looking very promising at this point. Sorry, the second question. Yep.
Yeah, thanks very much for the really fascinating presentation. So I live in Bangkok, And one of the trends that's very evident there is that the traditional urban home garden, backyard garden, is disappearing and is being replaced by condos and apartments and rooftop gardens. So my question is there any evidence about the likelihood of those rooftop gardens also having beneficial microbiomes? Because for an increasing number of people in cities like Bangkok, those sorts of rooftop gardens are going to be their main exposure to green space.
Absolutely. So whether it's rooftop gardens or community gardens, getting out into green spaces is the key part. And I think that we know that there's a tremendous amount of microbial diversity in soils, and that often gets aerated. And so my take home would be absolutely, whether it's a rooftop garden, whether it's a place that it's your backyard, or whether it's a commuter path through a public park. I think all of those types of green spaces should be considered here.
I guess my concern is that a rooftop garden is going to be so different from a garden at ground level with deep soils and much more intact micro ecosystems. And a rooftop garden is just so artificial, and you're probably talking about 2-3 feet of soil as opposed to 10, 20, 30 feet of soil.
And it probably relates more to a health question, so whether those rooftop gardens are healthy for us. And I might pass over to you there, Chris. So I'm not a health expert at all, and I don't really want to make strong suggestions as to whether it's going to be healthy or not, but generally a microbial diversity is going to help our immune system, so that's the important component. But
top 10, that's all you need.
Yeah, so I think that if it's a health-related question, I'd probably prefer to defer that over to one of these guys, if that's okay. But thanks.
Thank you for that. Look, I'm a geographer, not a health guy, but I've been working in public health for quite some time. I think the answer to that is we don't know. Right, so what we're going to do now is we've given you some information, what we think the challenges are. We've given you some of the science behind it, and now we're going to say final short video just to nail out what is exactly we're asking everybody to do.